Okay. So, hi everybody. Um, so today we're here to hear from Dr. Bueckle some interesting talks about visualization and virtual reality. So, Dr. Bueckle is the research lead in the cyber infrastructure for network science center at the Ludi School of Informatics, Computing and Engineering in Indiana, Bloomington. Is interested in information visualization, virtual reality, augmented reality, and is from Germany. He has a BA in media studies from Everard Karls University in Tübingen, MA in communication from Berlin University of Arts, PhD in information science from Indiana University. Andy developed a deep interest in digital artifacts, videography, and photography in particular. He worked as a video journalist and cameraman on projects in Germany, France, India, and the US. And then he decided to switch to more technical education and start to push on finishing a PhD in information science under Professor Katie Borner from Indiana Bloomington. Andy has a TED talk titled Living and Learning the Metaverse, which you can view on YouTube, and I think I shared with you the link, and on the TED website. You can learn more about his research on and creative work as well, so photography and stuff like that, on his website. I think you might have already the links to these references. And I'm gonna stop stealing time from Andy, who will be talking about building and exploring the human reference atlas with virtual reality. Thank you, Andy, for the opportunity to have you in the seminar series.
Thank you very much, Alessandro, for the kind invitation. I want to send warm greetings up to Fort Wayne. I'm here in Bloomington, so about 3 hours southwest. We just had the eclipse 2 days ago. I assume you guys might have had it too. So there was quite a lot of traffic here. So it's great being invited today to talk to you about building and exploring the Human Reference Atlas with virtual reality. Again, my name's Andy. I'm research lead here in one of the engineering labs at IU. So there is a vision in medicine for the usage of virtual reality and augmented reality and these associated kind of immersive techniques for use with patients. So what you see here is very clearly a stock image. You see two people, probably nurses or surgeons, looking at an organ here, kind of just suspended in air, kind of like a hologram, probably pre-operation. Of course, if we look just a little bit closer and wonder ourselves, what are they actually doing here? We realize that this is definitely fake. It is just an image illustrating an idea. We do not see any major data overlays here that would illuminate how this would be interesting for these two surgeons to look at. We would also assume that this kind of planning happens way before a procedure actually happens. So there is a vision for using these immersive technologies for very concrete patient care, but the reality currently is a bit different. So most VR and virtual and augmented reality applications exist for learning anatomy and physiology. You can see that here on the top left. And of course, VR and AR lend themselves very nicely to that because it allows you to take a human body, any human body, really. You don't have to actually procure an actual human cadaver and with everything that comes with it. And then you can disassemble it. You can just ignore the laws of gravity and just, pry everything apart. And then, these models vary in detail from a bit more photorealistic to low level of detail or LOD. You can see this one is a bit more illustrative. This is the musculoskeletal system. And here you can see a slightly more, quote unquote, realistic version. You have some specular highlights here, and it also looks creepier, of course, because it is more realistic. But none of these existing applications display any kind of patient data normally, and definitely not in large numbers. So this is purely anatomy focused and focused on teaching medical and nursing students basic human anatomy. So this is where I want to introduce or reintroduce the concept of the information-rich virtual environment. This is a term that was coined by Bauman and his co-authors in this really seminal 2003 paper, where they write, an information-rich virtual environment is a realistic virtual environment that is enhanced with the addition of related abstract information. So the idea here is that we would take a three-dimensional environment, which is spatial by nature, and then we would add abstract two-dimensional information that we would normally see in data visualizations. So for example, on the left side, this is from their paper. This is from 2003, so 21 years old. This is a throat And in the background, so you can see the tongue here in the foreground. You can see arteries and veins here in the back in blue and red. And then here you can see the palatine tonsils. You can see that here. If you ever had an inflammation, you know exactly where those are. So here we have a close-up of this palatine tonsil. You can see there's a bit of a texture here. And then we have this overlay 2D field that says it has a categorical label, which is, a text label. It's the right palatine tonsil. And it also tells you the surface area, 295 square centimeters. Full surface area for this. And then likewise here on the right side, this is also from that paper, we have a browser where we have a series of geometric primitives, so a rhombus and a cylinder and a cube. And it just overlays, again, these two-dimensional information overlays that tell you some numerical information that is not really spatial, but that relates to this semantic object. We're going to come back to this here in just a second. So I want to now talk a bit for the next 20 minutes or so about the Human Reference Atlas, which is featured pretty prominently in the title. This is what we want to use VR for, so that we can explore and build it. So the Human Reference Atlas is a comprehensive high-resolution and 3D atlas of all of these cells of the human body. So that's 37 trillion cells, and of course across also this has to account for 6 billion individuals that are living currently. So it's an immeasurable, like unimaginable large number of diversity. It is funded currently by 5+ scientific consortia. And the HRA at its base defines the 3D shape and space of anatomical structures in the body. Think of individual parts of the heart, individual parts of the brain, individual parts of the lung that all have distinct functions. It further defines the cell types of biomedical relevance that are usually found in these anatomical structures. As you can imagine, your left atrium in the heart is going to have a different cell type population from the cortex of your right kidney. And then, of course, that varies considerably by age, race, sex, etc. And then finally, it also maps the biomarkers that are used to characterize these cell types. So cell types express a variety of genes and lipids and metabolites. And so the Human Reference Atlas tries to characterize these biomarkers as well. And then finally, and this is the computational part, anatomical structures, cell types, and biomarkers are validated and presented into ontologies. And ontologies, for those of you that are not aware, are knowledge representations that assign IDs to unique items of knowledge. So there's ontologies for genes, ontologies for disease, ontologies for shopping products at Amazon. All of these can be organized with ontologies. And of course, there's also ontologies for anatomical structures. So these are some of the ontologies that we're using. We're going to get into that later. The HRA also defines how new data sets can be mapped into the HRA. So that can be done either spatially using these common coordinate frameworks, which we will cover later, or via ASCT plus B tables, that's anatomical structures, cell types, and biomarkers, ASCT plus B, ontology terms and IDs, or via gene and protein expression data. That's quite a lot. We're going to handle each of these individually over the course of the talk. Now, importantly, again, this is where it becomes computational. The HRA is provided as linked open data. In spatial, in that we indicate where in the body a certain tissue registration or an organ or an anatomical structure is. Biological structure, that means which cell types can typically be found in these organs. And then also linking this to specimen and clinical metadata, so the donor age, sex, etc. Everything that we build into the HRA comes from an individual, from a person that has a set of demographic metadata associated with them. And of course, those are captured as well. And so over time, we're going to be able to tell differences by individual demographic markers, such as sex, age, race, etc. Now, the HRA is authoritative in that there exists expert agreement and that all of this is validated by data. Our lab, we have one biologist on the team. The rest of us are all computational. So we're working extensively with specific organ experts across multiple teams in the US and in Europe. And it is incredible just how much specification can happen for just one organ. It is, of course, also computable in that it supports API queries and user interfaces. So the HRA data is not in one big spreadsheet somewhere, but rather it is organized as a graph that can be queried with query languages or via an API, and that also has user interfaces for exploration without any coding skills. It is published as I said before, linked open data, LOD. So it means it's connected to gene, disease, and other ontologies and data, meaning it can be integrated into the wider realm of knowledge out there. It is also open in that anyone can use the data and code. There is no paywall anywhere. And it is also continuously evolving. For example, new technologies might become available, new assay types, new organs might be added. We have a release twice a year. So in June and December is when we release the new versions of the HRA. Next version release will be in June, HRA 2.1. Now, I want to hone in a little more on the items that are relevant for virtual reality. So major components of the HRA 2.0, we have 65 anatomically correct 3D reference objects for 29 organs. So most of these are male and female, some like the placenta, or the ovaries are only for one of the two sexes currently. We also have 33 anatomical structure, cell types, and biomarker tables, ASCT plus B tables. And then soon we're also going to have cell type populations in 3D. That's like the latest and greatest thing that we're working on here. I want to focus on 4 HRA applications. There are more, but these are the ones that I want to cover here. The registration user interface, the exploration user interface, these two, I like to call them sister interfaces because they are related in their function. The ASCT plus B reporter, and then the HRA organ gallery in virtual reality. That's the virtual reality bit. There's a series of publications here. I'm going to put those into the chat later. So I want to talk a bit about the spatial data that goes into the HRA first. And of course, the meat of the matter is that to build this HRA, we need to register tissue blocks from diverse donors into so-called reference organs, which, as you can imagine, is not an easy feat. Everybody's kidney, for example, you can see that this is a part of a kidney here on the right. Everybody's kidney is different. Some are larger, some are smaller. Others have, a certain number of renal pyramids. That's what you see here. Others have, some have 7, some have 6, some have 8. So we have to work with reference organs that represent a huge variety of human diversity. So, but ultimately, these cell type populations, which you see visualized here in a more artistic way, will contribute to cell type populations for entire anatomical structures. So going from individual tissue donations from humans to averaging and summing up cell type populations for entire organs and entire anatomical structures. So the question is, how do we register these tissue blocks? And so the problem is that documenting tissue extraction sites is non-trivial. You can see on the left side, this is the best case scenario. In the best case scenario, there exists a photo of the entire organ. This is a dissected kidney. You can see the outer kidney here with the cortex. You can see the renal pyramids nested in here. You can see some fat deposits here. And then this is a chunk that was cut out of that kidney. You can see some of the renal fat here in the middle. But of course, most of the time, the people that register these tissue blocks do not have this kind of material because, if the surgery is done in a hospital in New York and then the tissue is shipped to Vanderbilt University and then the registration is done at the University of Michigan, that's already 2 degrees away from the surgery room, and then this information might have been lost. Or at least the photos might have been lost. There might be a textual description left of where it comes from. So these photos, if they're available, are rare. So we use the visible human male-female data set, which is linked here, to model the 65 3D reference organs. And so this is one of them. This is the male left kidney. So we built a registration system where you have a virtual kidney, and then you can indicate here with this little block where exactly the tissue block was taken from. So in action, this is what the production version looks like. I prototyped the original version as part of my PhD. You can see here the user is taking a cube. This is all running in a browser right here. And then the user can drag the tissue block into this reference organ. Again, this is the male left kidney. And then they can just move it here with their keyboard into position. Now, the sister interface, the registration user interface, is the exploration user interface. Here you can see all of the registrations, so 730 public ones that have been made as of whenever I took this GIF, which was, I think, a few days ago. So you can see here all of these organs and how many tissue blocks are already in there. You can also pretty quickly see which parts of the body have been mapped well and which ones are lacking. So the kidney, of course, has gotten a ton of attention. Back here you see the spleen that also has a ton of attention, but then there's large swaths of skin that have not been mapped. Of course, we have reason to assume that the skin doesn't change quite as much depending on where in the body you are, but this is a very quick and nice overview of, which parts of the body still need more mapping. So I mentioned earlier the concept of the common coordinate framework, and it's important to understand that these bodies and organs that we're looking at, they are nested inside of each other, inside of this common coordinate framework. So a CCF is, for the human body, provides a unique address for each cell in the human body. It's kind of similar to a latitude-longitude system that you would use to navigate a world map. Of course, the CCF has to be 3D and multi-scale. You've already seen kind of the 3D part. All of these organs are in 3D. These interfaces are in 3D. So let's talk about the multi-scale part. So as of the HRA 2.0, we have 1,218 anatomical structures in the male and female bodies. So then what we can do is we can go steps down. So we see a user here. This user happens to wear a VR headset standing next to the visible human female. That's the female donor. You can see in the middle the blood vasculature with a bunch of organs. And then, suddenly we go down to the organ level. So this is one kidney. And then we can get shrunk down even further into a renal corpuscle, which is it's a functional tissue unit that helps with the filtration of toxins from blood. And then we can go down even further to a podocyte, which is one cell. This is one cell. Then we can go down even further to a gene, NPHS1, which maintains structural integrity of the filtration barrier. So this is the multi-scale nature of the Human Reference Atlas. Now, shown this in a more geometric way, we can see that these reference systems would be nested into each other. So we have one gigantic atlas reference system at the root. And then in there we have two bodies, a male and a female body. We really need a pregnant reference body as well, but that is not currently within the scope. But you will see later why. And then inside of each body, we have about 29 to 30 individual organs. So in this case, for example, the left kidney in the female reference body. And all of these have their own 3D reference system. And then inside of each organ, you have a Z-stack, you have a tissue block. So there's like one block of tissue, that's one of these cubes that you saw in 3D earlier. And these again are cut up along the Z-axis into individual tissue sections. Now each of these sections can then be segmented, and then we actually have the original cell. So this allows us to create a three-dimensional address for each cell going along this multi-scale kind of dimension of the Human Reference Atlas. So this allows us to 3D register as well as explore tissue blocks spatially and semantically across the macro, meso, and micro scale. So we talked about spatial. Let's now talk about the biological structure. This again where it becomes a bit more computational. So I already mentioned the anatomical structure, cell types plus biomarker tables, or AST plus B tables. But what does an AST plus B table do, and why is it relevant for us? So unstructured knowledge sources make up about 80% of biomedical knowledge. That's an insane number if you think about it. So this means that this knowledge is encoded in papers that you have to read, humanly read to extract the knowledge from it. But AST plus B tables standardize how information specifically about cell types and anatomical structures and their biomarkers is captured, formatted, and labeled. They thus capture knowledge about organs, AS, CTs, and Bs that uniquely define cell types. And again, they connect these units of knowledge to ontologies, like for example, Uberon, that's an ontology for anatomy, and also cell ontology, that's an ontology for, cells. And so this way, AST plus B tables provide nomenclature, synonyms, descriptions, relationships between different entities, and they provide what's called provenance for knowledge. So a trail of understanding how knowledge was made and how it was created. And of course, each AST plus B table is written and validated and authored and reviewed by organ experts. And then we can assign unique IDs for each unit of knowledge. That's kind of like also the base idea of ontologies. So, of course, these tables become insanely big and they become very hard to read, nearly impossible to read. So we built a tool called the AST plus B reporter that visualizes each of these tables as a tree. So in this table here, in red, we have anatomical structures. In blue, we have cell types, and then in green, we have biomarkers. So in this case, for example, this is for the heart. So the heart has an anatomical structure called the right atrium. And the right atrium consists of three subanatomical structures, the sinoatrial node, the myocardium, and the vasculature that feeds into the atrium. And then you can see these are the cell types, 8 of them to be precise, that are typically found in the vasculature. And then here linked, we have the biomarkers, the genes in this case, that are typically used to characterize these cells. So these cells express these biomarkers to a heightened degree. And this allows us to visualize all 33 anatomical structures, cell types, and biomarkers tables that we currently have. So to tie this back together with the spatial data that I mentioned earlier, now we see the lung here as an example. This is from our 3D reference library, which is also available for free. You can browse all of the organs there and download them. You can see here in this view, there's a bounding box around each individual anatomical structure. And the beauty then is that we can tag each of these anatomical structures with an ontology ID. So we can tag it with corresponds to unit X in this and this ontology. And so this way, when we talk about, for example, the anterior left lobe of the lung, we always talk about the same kind of entity, the same kind of term. So these organs have been custom built by our medical illustrator team with input, of course, always from subject matter experts. These organs support the RUI and EUI, which I was already showing, and the HRA organ gallery, which I will show here in a sec. And it also allows us, again, to label anatomical structures with ontology IDs. So that's all nice and well, but what is the HRA actually used for? So for that, we have the HRA user stories. We defined a total of 7 of them for the HRA. We have also documented that on the HRA portal. And I want to note here that this section demonstrates codes under active development, so the UI will be improved. So HRA user story number 1 is to improve cell type annotations. So the goal here is to use our API to improve the accuracy of annotations for bulk and spatial data. So biological single cell data. The input here is that you define the spatial location of a tissue block, and the output is you get a predicted cell type population for it. This can be used for a variety of things. For example, to do quality assurance and quality control. Let's say you have a piece of tissue, you have analyzed it, and you get a cell type population back. You can run it against the HRA to see if that matches the expected cell type population. Likewise, if you have not done the assay type yet, you can get a preview of what you're likely going to get. So the way this works is on the left side here, you can define an extraction site. So you will see here, this is the registration user interface again that I was showing. So here I'm choosing an organ. I'm going to go here for the female left kidney. And then so here it is loaded. And then I'm dragging this cube up here. So this is the site that I'm interested in. And then this is what you get back. It's a long table, essentially, that lists all of the different cell types with their percentages and numbers that you are likely going to get. So that's user story 1. User story 2 is to predict the 3D spatial origin of tissue samples. So here the goal is to use biomarker expression values to predict the 3-dimensional location of tissue samples. As I mentioned earlier, oftentimes the exact spatial origin of tissue is lost in the process because it was not deemed relevant to capture it at any significant level of detail, that goes beyond just the organ and whether it was left or right. So the input here is a cell type population of a tissue block, so it's the inverse case from earlier. And the output are 3 things. We're kind of giving a goodie bag of things. So first we give a list of the anatomical structures with the most similar cell type populations. We give a list of the most similar existing data sets that are high quality, that are in the HRA, and also the locations of the tissue blocks with the most similar cell type populations. You can see that visualized here. And then kind of as a bonus, you get a complex 3-dimensional corridor that was made from one spatial location that encompasses all of the possible locations with the same kind of cellular makeup. So again, up here, you input a CSV file. In this case, I'm just using a sample CSV from the heart. And then you get back a list of similar data sets. You get back this 3D visualization of the most similar tissue blocks that have already been registered, and a predicted list of anatomical structures that have similar cell type populations. So having talked about how it is used, let's talk about the 4th HRA application that I was teasing earlier, and that's the HRA organ gallery in VR. So as I was showing earlier, the exploration user interface allows you to zoom into the body in your browser, move your orbit camera, click on tissue blocks, and understand what has been registered where and who registered it, and look at all of this together in context. But of course, this is always going to be limited to the confines of a 2D browser. So we were wondering, what if instead of browsing the Human Reference Atlas at the 3-dimensional level on a computer where you're limited to your browser with your mouse, what if you could instead stand right next to it and see these organs in their real scale? Those have been modeled, to mirror the actual scale. So if so if a user stands next to the visible human female, they appear at the correct size. And so the HRA Organ Gallery in VR is now for free available on the MetaQuest store. If you have a MetaQuest, feel free to scan this code here and download it for yourself. It's again free of charge. It's also a paper that we published earlier last year. I'm currently working on a follow-up one for it. So this is what the HRA Organ Gallery looks like. Of course, it's always kind of awkward to demo VR things over WebEx or Zoom, but this should hopefully do the trick. So you can see the two humans here in the middle. You will see that it's the same bodies that you saw earlier in the Exploration user interface, except we gave them a different color for aesthetic reasons. They now look more like marble sculptures. The blue was really just a compromise. We didn't want to assign an explicit skin color. Then in the front here, you can see this app is supposed to be used while sitting. So the user sits at this gigantic keyboard, kind of like in an airplane, and then they can click on one of these buttons, and then they can order an organ into this so-called grab cylinder. And that then allows you to move and grab and rotate and scale the organ and investigate it in its 3D spatiality. At the same time, we have this outline here that shows, so this is the female heart. This then shows where the organ actually sits inside of the main body. We have this monitor here on the right side that shows you some metadata. So we currently have 730 tissue blocks in the scene total. The selected organ is the heart, and we have 73 tissue blocks nested in this organ here. This is what it looks like in your usage context. So here we have a human user standing next to the table in this case, and just using this ray interactor here to play with the heart. This is the view from the top. You can see we have this grid floor here. It starts at a 3 foot by 3 foot, so a meter by a meter for each of these tiles. And then as the user travels across the CCF scales, they're going to rescale. That's one thing that we're currently implementing. As I mentioned, we have a keyboard for organ selection. So the user can pick any of these organs that are available here. We added, of course, the names for each under the icons. They can also reset the organ. They can choose the sex and the laterality if applicable. So, of course, the heart exists for male and female, but there's no left and right heart, so those are currently disabled. And then we are currently working on implementing different usage modes, which I will hopefully be able to demonstrate in a future talk. As I mentioned earlier, we did use custom shaders to improve the aesthetics. As you can see, this whole thing kind of goes for a clean room kind of aesthetic, very abstract. Of course, this is something that you normally don't have to worry about if you develop two-dimensional applications in browsers. There is no 3D space to worry about. We have to worry about, okay, how can our user actually move through the scene? How can we reduce motion sickness? And how can we make the whole environment conducive to the aesthetic experience, while of course also make sure that it runs reliably at 60 frames per second or more on MetaQuest headsets. So the maximum you can use for a MetaQuest 2, which is what we're targeting here, is 1 million triangles. And if you show all of the organs at once, you get about 4.5 million. So there are tricks that you have to apply in order to, so we can never show all of the organs at once, for example. We also cannot use any post-processing or high dynamic range. But those are just the limitations of the hardware that we're working with. We recently bought the Apple Vision Pro, and we'll see what kind of performance improvements are needed for that one. It is a lot more powerful, needless to say. Another thing that's in progress is a three-dimensional monitor here to display metadata. As I mentioned earlier, the tissue blocks, the organs, and the organ blocks that are currently in the selected organ. These are, one of my students made this neat little annotation that is shown to the user in VR. It shows how to use the controllers to achieve the kind of controls that they want. It's a relatively simple control scheme, but if you've ever used VR before, you know that it takes some time to get used. What I typically tell people when I put them in VR is, I ask them if they've ever played a PlayStation or Xbox game, then I tell them that the controller works basically the same way, it's just kind of cut in two. And of course, the user uses these controllers to locomote, so to move around and to interact with the organs, and that takes some practice. This is another screenshot that showcases how organs are selected. So the user intersects with the button and then presses one, and then the organ is loaded. This proved incredibly hard to do for people for some reason, so we might have to refurbish that one. This is actually what we're going to do next. We're going to use, this is a so-called ray interactor that allows you to just point at something that's farther away and then select it that way. Here's a screenshot of a user being in VR and having rescaled the organ to about 10 or 20 times its normal size. So the normal heart is about 10 centimeters tall. So that's about 25 5 inches, something like that. So of course this is much bigger. This is like taller than a person now. But all of this is possible here in our VR application. So how do we bring in the data? Like how does the HRA data come into VR here? So as I mentioned before, we have an HRA API, just documented here. And this API has endpoints for individual organs, tissue blocks, and our entire scene. So if you were to build your own 3D application with HRA data, you could absolutely do this by just using this the scene endpoint here. So this is what the response looks like when you go to it in the browser. This is a specification for a 3D reference organ for the skin, actually. So it comes with a series of values. The important one here is the scene graph. As you can see, this is just a download link. This takes you to a website where you can download the actual 3D object that is the skin. So you could build an entire 3D scene just with one API call. So that's kind of what we're doing here. So as I mentioned before, we are using the scene endpoint, which provides all 65 organs on our 700 plus published tissue blocks with all of the metadata. So what we then do in our application, we're using Unity, which is a video game engine. We preload the organs, and then at runtime, we create these organ data components that capture all of this metadata that you saw earlier in the API response. And because the video game engine is basically just like having a player or an enemy character in a video game that has specific properties that we can then edit at runtime. We can color them based on that on runtime. We can allow the user to filter by a specific metadata field, etc., etc. And then likewise, the organ itself, we can annotate with these tissue blocks here. These are created at runtime, which goes incredibly fast. So we can create all 700 or 800 or even 1,000 at some point with very fast speed, and then we can also capture the metadata for those as well. So looping this back around to the beginning, I was talking about Doug Bauman's concept of the information-rich virtual environment, and the beauty there was that this offered an early idea of what it could look like if you have an environment where you have spatial 3D models that are enriched with 2D information. So now 21 years later, we're looking at the tonsils again. So remember, this was the tonsil from Bauman's example. Well, these are the tonsils in the HRA organ gallery. You can see them here nested. You can, of course, see they have a much higher resolution now. They have better lighting because graphics have just become so much better. But it fully implements this original vision of the IRVE that Bauman drew. And then likewise, we had these semantic objects back there that, geometric primitives that are annotated with rich metadata. And we have the same thing now with these tissue blocks. Each of these tissue blocks has all of the information in it for the donor age, sex. We can link cell type populations to it at some point, linked publications, and all of that makes up this big linked open data aspect of the HRA, and now also in VR. So in terms of next steps for the organ gallery, we will of course expand its functionality. We received an NIH Jumpstart Award last year with our collaborator Lu Shen, where we held a 2-day hackathon at NIAD. You can see us here. This was, of course, a really special event because if you are into virtual reality at all, this was absolute heaven. There were so many VR headsets there. So we just got together with virtual reality experts across NIAD to prototype and hack the HRA organ gallery. And so we achieved the collaborative performance and usability improvements. We increased the mean frame rates per second from 30 to 65. Very important to prevent motion sickness. And also important to actually get this into the MetaQuest store. They are very particular about performance because of the restrictions that the MetaQuest has. We were able to reduce the number of triangles from 4.2 million to about 930K. We did that by applying algorithms to decimate the number of polygons needed for all of these organs. We also improved the usability of the organ selection keyboard. So since March 2023, we conducted user analysis with 20 plus experts from 4 plus consortia. You can see me here, talking to a whole bunch of different folks at different events. And so as you can tell, I am quite passionate about ensuring that we get expert feedback and buy-in into this application. I'm an information scientist and my background is in media production. I do not have biological expertise. So our whole team relies on the input from experts on all of these individual particular organs. And so having these kind of continued discussions is essential. And one of the advantages that we have is that VR is still so new and so promising that most people are very happy to provide their input, which is great. So far we identified 3 major use cases. We identified quality assurance and quality control. So going into the organ gallery, making sure that maybe our tissue registration is right, making sure it's not floating around in space somewhere. We actually already identified a few tissue blocks that way that were just not in the right organ, but they looked as if they were in the right organ. Onboarding to the HRA, so an introduction to the Human Reference Atlas 3D DOs, like the 3D digital objects, that's the organs. And of course, also this is probably the most enticing one, but also the least developed one. That's the telling embodied data stories. So telling stories about what a healthy reference looks like versus what a diseased donor would look like. Of course, this is a bit more touchy, and this is also not fully developed yet, but this is one of the use cases that we identified. So we need further refinement with expert input. My next goals are to identify and implement pilot projects for extending the HRA organ gallery. And of course, to also build a strong network of collaborators that are interested in the research and application of VR, which is why I'm happy to be speaking to all of you today. I always like to show this bit. When we had finished this hackathon, we were finally able to tick this off of our list in Asana. And this was, of course, a really important milestone for us. And we are currently envisioning hosting a second of these workshops this October, again at NIAD. This was, of course, all a collaborative effort. You can see a variety of folks from NIAD were included in this. Okay, with that, I want to go over to acknowledgements. I want to thank my excellent team at IU, including my PI, Kadi Borner, and also Lucien from Stony Brook University, who was working with me on this Jumpstart Award. This is the acknowledgement that we have for the Human Reference Atlas. I'm going to put this link to the HRA portal here into the chat in just a second. Here are related papers, and I would love to stay in touch. I think we have some time left for a Q&A. I would love it if you could reach out if you have ideas for collaboration. And that is the end of my talk. Thank you.
Awesome. Thank you very much, Andy, for the great presentation. So we have some time for questions. The priority goes to the students who are currently helped of the Data Club, currently helped organizing and sponsoring the event, which are on campus, and I hope they're being able to follow along. And so, any questions? There is need to break the ice. I don't know how to say it. I'll start. So I have a question about yes, Professor Kim. Yes, please.
Can you hear me now?
Yes, very well.
Okay, so it is a great presentation. It's very interesting project. And my first question is, as a kind of a researcher studying in applying or using VR to be a practical way. So I tried a couple of projects. One of my main difficulties, if I understand the terminology correctly, then what that is called it a disembodiment problem. So easily speaking, what we have in the real space versus the virtual space is what different. So my previous participant commonly experienced difficulties either grabbing object in the virtual space and interact with especially the object which is what either rescaled using the scaling factor. So does your team have any plan how we can address the common issue using a virtual reality in real world?
So that's an excellent point. The answer is yes, we are having some strategy. So one of the problems, for example, that we have is that we put people into VR, they see the organs, they're fascinated by it, and then we tell them to press a button on the keyboard. And even people that I expected would be able to grasp this concept very fast do not know how. So if you tell them just touch the button with your hand and press it by pressing a button, most people cannot make this leap for some reason. So I think one thing that really helps in VR is if people don't have to move to something, but rather if they can fetch something to come to them. So I was mentioning earlier, let me go to this part of the talk again. I was mentioning earlier the idea that people had trouble pressing the keyboard. I had an illustration for this. Here we go. Let me here, and let me share my screen again. Here we go. So can you see my screen again, Alessandro?
Yes.
Yes.
Yeah,
Okay,
yeah,
thank you.
yeah.
Okay. All right, cool. So I mentioned earlier, people have trouble making this kind of logical step, like that they have to move their hand onto the keyboard. So instead, we can kind of make the keyboard come to them by giving them this kind of magical wand or ray interactor. I wanted to avoid having this because it introduces another graphical element that users have to understand, but it bridges that problem. Another problem we found here was that, if a user, let's see, if the user typically sits here, like the user sits in real world physical space, they sit on a chair, and then usually they sit about this far away. So if I ask them to press a keyboard button, they move their hands forward and they just won't reach because for some reason most people do have they cannot understand spatially how far they're actually away. So giving them this ray interactor will help them bridge that distance. So I think the answer to the problem lies in conscious design decisions. Looking also at, of course, what video games, for example, obviously we're very inspired by video games for VR that have been put out now that have these kinds of issues all the time. And so I think there's a lot that we can learn from that on the design level. I hope that answered your question.
Yeah, yes, it is. So basically, your team tried to help the user to be more familiar in a virtual environment through some kind of systematic training or coaching.
Yeah, we have it. Well, currently we just have annotations that show the user what they can do. Of course, normally when we demo this, I'm right next to it, so as an embedded researcher. We have a long-standing GitHub issue to create a click-through tutorial. What you would have in a video game also, where you don't have a game designer standing next to explain to you what to do. You have to learn it yourself with the tutorial. So we're currently working on implementing a tutorial, which if you've ever done that, it's very hard to do that in a way that anyone can just use. It's hard enough if it's on a two-dimensional screen. If it's in VR, it's even harder. Of course, we can assume when people do use the application on their own, if they download it from MetaQuest store, they probably already have some experience in VR, proven by the fact that they actually own that device and that they went there through the trouble of installing it. But yeah, hopefully in a follow-up talk, I can introduce the tutorial that we're currently cooking up.
Great, thanks.
Awesome. Other questions?
I don't have any questions, but I really enjoyed the presentation. I've been looking through the articles and very, very impressed, very impressed.
Thank you.
So I have a couple of questions on, let's say two questions. Okay. So when people do brain imaging, they have maybe some sort of templates that they use to register the brain to some standard system. Sometimes it's spatial, some other times it's something a little bit more complicated. So then usually they have maybe 100 or something like order of 10 power to maybe individuals, and they use some sort of average in some carefully done way to get that template as valid as possible. Okay, for most general population. So how do you address that? The same type of matter. So how do you do that? Because there it's as you explained, it's multi-scale. It's not simply pixel by pixel, but there is something extra to do that. Maybe, yeah.
Yeah. So at present, the brain, just like all of the other organs, there is a gross anatomical registration, which is just a cube, right? And then depending on which portal the actual data is from, we link to the MRI data, or we link to any CT scans or any H&E stains that are lying somewhere. So this is basically just a hub that for now that links you to all of the to where the user can actually find the raw data. One of the things that we currently implement is the ability for the user to actually click on tissue blocks or pick tissue blocks and then get out the URL that leads you to the portal where the data comes from, so that you can do some more research outside of the virtual reality organ gallery. One thing I want to show here that's actually going to be interesting here, since you mentioned the brain. This is from a recent team update. The brain, so what you see here is a screenshot from the organ gallery. You can see this is the male here in the background. You can see that this is the brain that is selected here and then outlined. What you will notice here is that the brain has a really funny shape. The reason for that is the brain that we are using comes from the Allen Institute, and it was never optimized for VR or web browsers for that matter. So it has so many triangles that it instantly crashes the application. Like, it goes down to under 1 frame per second and it becomes unusable. So we had to cut out all of the parts of the brain for which we don't have any registrations. You can see in here, kind of nested in there. See if we can zoom in, and yeah, you can see some tissue blocks that are in here that have been registered. So we wrote a script that identified which of the brain parts have registrations in them, and so we only kept those. That's currently where the multi-scale part of the organ gallery stops. But bringing in more scales so that the user can travel down to a renal corpuscle or individual cells, that's currently in preparation. We already have some 3D models ready to go, but that's in progress.
And the other question I have is, so how many atlases do you have? So what I'm wondering is, do you have something which works specifically for some subpopulations? Okay, not something like it could be, yeah, that's a question, right? So yeah.
Okay, so the way I understand it, you're asking about cell type populations. We currently have cell type populations for 74 anatomical structures in 13 organs. Mostly those are in the heart, the lungs, and the cell, and the kidney, for two reasons. One, this is where we have the most tissue registrations, but this is also where we have the most single cell data. We are trying to add more organ as we go along. In order to construct a cell type population, we need three things. We need a registration with the RUI, which is what I showed today pretty prominently. Yeah, like, you need a 3-dimensional cube that is placed into one of the organs. You need a cell type population for the data set that has been derived with a cell type annotation tool. And you need this, it needs to be scientifically validated. So it either has to be associated with a peer-reviewed publication, or it has to come from a single cell portal that has quality assurance and quality control. For example, the HubMap portal. So far, we have that for 553 data sets, for 74 anatomical structures and 13 organs. So it is growing. And we're currently preparing a paper. A preprint should be out hopefully in the next 2 months, that introduces the workflows and the original data set that contains all of these cell type populations. And of course, the ultimate idea is that the organ gallery is always downstream. So the HRA is being developed and grown, and new organs are being modeled. And then the organ gallery is an application that uses the HRA. So the organ gallery is always downstream. So when we develop something on the cell type population front, it's going to make its way through the API down into VR. So that's where we're currently at. We're trying to build the cell type populations, and then later on, we're going to show them in there.
Right. Thank you. Other questions? Yes, Professor Kim.
Okay, if there, I do not want to use too much of other person time, but if there is no question, actually I have one more of a curiosity. So right now, majority of your application is somewhat processed in advance, so it's a kind of offline or processing. But in the long run, with the advancement of our computing, there may be some potential of we may able to reconstruct at least a part of the body in real time. So my question is, in order to reconstruct, for example, I'm very interested in converting into my CT scan into the 3D volumetric space. So right now, what is a kind of in general, a kind of a data processing time, and what kind of computing power does your team use it?
Yeah. So in order to construct these 3D reference objects, we also use CT scans and MRIs from the visible human data set. The visible human, for those who are not aware, is a data set from the 90s from an individual who donated their body to science. And then after his death, for the visible human male, they cut him up into hundreds of little slices that are available as CT scans. So what we did is we took these CT scans for individual organs and then we traced them using 3D slicer, which you're probably familiar with, and then generated a volumetric 3D model from there. Of course, only at the anatomical precision required for this kind of gross anatomical tissue registration. The organ gallery itself does not do any volumetric generation. The organs are modeled and then released as part of an HRA release, and then they're being used in the application. The way that we generated these models was with medical illustrators that used, again, 3D slicer to generate this kind of model. I'm going to put the link to our 3D reference object library into the chat because that illustrates really nicely which organs are available. It also tells you a little bit about the process for the entire thing. But that's an excellent question. And as I mentioned earlier, with regards to performance restrictions, we are targeting the Meta Quest 3, but I recently purchased the Apple Vision Pro. And I'm currently in the process of just building some small Hello World, or as I like to call them, Hello HRA applications, where we can just do some benchmarking with regards to, okay, how big is the difference, for example, in frames per second and the level of like VSync issues. So it's another thing that we're working on. Yes, I think there was another question.
Professor Cracker, I think.
You were talking about instructions and how difficult that was with the instructions, and you may have to outsource to experts who actually do this professionally. I had to do this. I'm a subject matter expert for national organizations. I'm a specialist, and we have had to do that when we have created this kind of instruction. We've had to outsource it to specialists, and it is much easier to do that.
With instructions, you mean instructions on how to use the VR application?
Yeah, that you were talking about the manual earlier in your discussion. We were talking about the manual, how difficult it was. And I understand that. I mean, I've had to work weekly with experts on that just to do basics, just basic instructions. So we've had to outsource it, like for the American Dental Association, we've had to do that.
Okay, yeah, that's a good point. Currently, we have a manual in the form of a slide deck that just has some screenshots from inside of VR. The problem with that, of course, is that a manual is not really practical while you're in your VR headset. So yeah, that's an excellent point.
Awesome. So maybe you have time for one last question. Well, maybe I'll thank the Data Hub Club and advisor Professor Dubois who helped organizing and sponsoring the event. And then I also want to thank Andy for the great presentation, and it was a lot of fun. And thank you very much for your kind availability of speaking in our seminar series. So thank you very much.
Thank you, Alessandro, for the invite. Thanks for all the excellent questions, and thanks to the Data Science Hub
Yeah.
Thank you for inviting me and hosting everything. Thank you.
Awesome. So thank you very much. Gonna stop the recording.